So the motivation behind this pilot is the same that is the ambition of Merck Healthcare overall. It's to bring more medicines to more patients and to do so faster. So we're often reminded that our patients just don't have the patience or the reason to wait for our slow, cumbersome processes, right? And for them, it's a lot more than just an inconvenience like it is for me. So if we were to go over a few buildings from here to one of our labs, where the lab automation is already taking place, you would see how process optimization and estimation has grown to the throughput of data generation of our machinery. So we have these equipment like this echo flip-mount sensor that use actual sound waves to sense nanoliter volumes of compounds with very precise volumes with very extreme precision and no contamination. And then we have the robot on rails, which move these tiny plates, 1,500 little experiments back and forth and transfer these plates from device to device and allow extremely high throughputs and extreme rates and volumes of data generation. Now, all of this, of course, comes at a price, and our lab automation has produced so much data that our scientists are now spending approximately half of their time in data wrangling, basically copy-pasting data from one application to another, collecting it at the instrument, curating it, reporting it, making sure the data fits to themselves. And of course, this is a big cost at the time to scientists who would rather be spending the time generating the data. Now, among this 50%, 30% is actually documenting the experiments they just performed. This is very often repetition of labor, since, let's be honest, that you have to enter the data, what is the experiment you're doing at the instrument, then the data is generated in digital manner, and now you have to report it to a different platform where the data is already digital anyway. So this is a waste of the time, and we'd like to return their focus to performing the assay and not wasting it and documenting the assays. Now, of course, in these times of high change, we want to be able to perform more with existing resources, and they can grow endlessly, and basically automating these data flows allows us to increase productivity by removing unnecessary steps and streamlining the way the data flows from the instrument to the database to the analysis itself. Now, of course, all of this automation comes at a cost. And we have the fortunate position here in healthcare sector that the time actually can saving time, can generate revenue. So it's been said that the daily revenue of a typical pharmaceutical during its patent exclusivity is something like $1.4 million, which means that if we can accelerate our cycles, these automations can actually pay for themselves. And this has the added benefit of getting the medicines to our patients faster. I'm sure to this audience, I don't need to preach to the choir about the value of fair data, but luckily now with the advent of AI and how it's becoming mainstream, I think even our management finally understands the value. Because in order to have the data be ready to train the AI algorithms, it has to be born fair. And in this definitely lets us justify the need for the fair data, I think even easier than before. Now, before I dive into demonstrating the actual lab of the future, I'll just briefly describe what it is that our department performs. So here is something called a DMT cycle. This is the engine of drug discovery. It consists of design, make, test, analyze, DMTA mix. Now, the part of the cycle that we are responsible for is test and analyze. And as you can imagine, it's an iterative process that loops over and over in an effort to make a compound ever better, ever more potent, ever safer, so that ultimately can become a medicine, a drug, a treatment. Now you can break down this test and analyze into actual physical steps of requesting testing, preparing the compound slate before the assay, and analyzing data. But of course, these are somewhat abstract and very generic, and in reality, there are many sub-steps that have to be formed on the way between requesting the test and actually getting the data to feed the cycle further. Now, because of the complexity of these steps and because the specialists that perform them have to have special domain knowledge, special skill sets, they're very often performed by different people, different scientists, with different specializations. And even though the individual sub-steps might take a few minutes, because of the fact that they come by different people, and because the instruments are robots on a rail, like our bioasset automation labs, like these handoff delays can add hours or even days to our process overall. Now, our goal is to find these steps that can be automated away, show them off into the system, to the cloud, right, someone else's computer, and allow the scientists to go back and bring their focus back to actually bring the data generation. Now, the process we decided to focus on is for this pilot called the Compound Distribution Hub QC. You might ask why we chose to pilot this. This is actually a very, very repetitive process where our scientists in our compound logistics and compound distribution hub have to generate the plates to compounds of different concentrations. It is a very structured process that is performed many times a day. And unlike the assays, which change from week to week, from experiment to experiment, it's a very standardized workflow. It is also truly boring, if I might say so, right? You have to copy paste data, you have to move the data from different instruments, generate the QC reports, and because of all these repetitive steps and how similar they are from the step you just did a minute ago, it's also prone to errors. And because the complication is unfortunately very hard to trace. Here is a diagram that kind of captures what actually is happening. So you have the mosaic to our compound business system. This basically generates a work order to our Momentum platform. This is a last automation bench liquid handler. And then this actually performs a compound plate generation, and then you get a bunch of different files about what just happened during the compound plate generation. Then these compound plates are evaluated using either mass spectrometer or echo, which evaluated the initial concentration. And then basically the scientist has to then form an Excel macro on this file, convert this file, property paste this, make a Excel document in the end, export the Excel document into a PDF, and finally paste this all into Pesto for the lab head to review and perform actual QC analysis. So what we did is we first looked at the individual files and we tried to see whether the necessary metadata and the actual data is there. As you can see here, I had in the box of the various types of data that has to be copied from the different files. Sometimes the metadata is in the file name, sometimes in the document itself. But what we did is we identified all these components, put them into a workflow that merges all the files together, and then wrote a Python script that actually processes data and generates a PDF report, which then can be placed into Pesto. And what this ends up looking like for the scientist, here I show in the video. This is a video from our development version of Pesto. Pesto is the Signals ELN interface here in the Merck talks there that Jörg will discuss a bit more later in the day. So here you see ELN entry being generated, and we make a table for the TDP work order, put in the plate numbers, and tell the Pesto to fetch the PDF codes. This takes usually a few seconds if it's 2 plates. If it's a few hundred plates, which sometimes on a busy day it can be, takes on order about 1 minute, which is not as bad as the formed workflow. And then here you can see what one of these GC reports actually looks like with the heat maps, the bar graphs, with demonstrating the compound concentrations, the BMSO concentrations, and contaminants, whether your dose response service, all with different visualizations of the lab it relies on for seeing the report. And here on top are the actual calculations whether this passes the various QC tests that are needed. Now, of course, a system like this brings new complexity to an already complex landscape. So here you see the different actual pieces of equipment that are feeding into it. The final aegis is an intermediate component which has to actually take the various files and upload it into the cloud platform. Then in the cloud, the data is processed, decorated with the necessary metadata, and the Python script performs the PDF generation. And then down here, you're seeing the PDF looks like. And then here we had another connector which plays the role of pushing the data from TetraScience into the Signals Notebook. Now, for now, we use a full process, but hopefully in the future we can maybe figure out a way to push the data into ELN. But if this is more of a process question rather than technical one. I'd like to here briefly point out the importance of visualizing the quality control. So our lab heads are used to looking at tens or hundreds of these every day, and they've trained their brains to be excellent QC machines, right? They look at the plate and they right away recognize what's happening here, and it was important for them for these visualizations to match what they had previously seen, and would help the scientists to be able to capture this what they did before. But of course, we have hopefully better visualization capability in Python than we did in Excel. So we are now working on improving on this now that they've accepted the old workflow, and that we can hopefully build on it and not just replicate with the manual workflow consisted of. So once the pilot was concluded, we decided to look at what are tangible results here, right? We don't want to automate for the sake of automation. First of all, the scientist side could be massively tight. So we benchmarked this versus the manual workflow and the automated one, and per 100 plates, which you can say happens on a daily or weekly basis, this saves something on the order of from 7 minutes to 10 minutes per 100 plates. The performance needs are also, of course, important. So we want to bring the medicines to the market faster. And unfortunately, when the process isn't so exciting, people often let the work pile up before it actually feeds the next set. And actually, when we looked at the historical data, sometimes this jobs were sitting around for up to 30 days before the scientists actually came and got around to the process of actually uploading the PDFs to the ELN notebook. And importantly, the error avoidance. So we basically quantified all the manual steps, all the potential sources of introducing error. And we decided that about 75% fewer potential on-track manual steps are in this process, basically 35% fewer sources of errors. Now, of course, now that this is solid was working, the question is what are future pilots? Now, having a successful pilot brings momentum, and because of our ambitions to accelerate the drug discovery process, we want to identify which workflows can benefit from these kinds of automations. But when doing so, I think it's important to avoid visual fatigue and over-automation. I think it's very easy to just randomly start picking processes that you want to automate away. And if you alienate the scientists in the process, and you very easily spend lots of money and in the end make both the scientists unhappy and our management as well. Now, based on this, I'd like to also show my recognition and gratitude for the people who actually did the work here. So here in the lab in search pharmacology, Dominic was the SME from the lab, and Zergona was the lab head. In the visual data and IT team, we have Tom, Kai, and Jörg, who are the heavy lifting on the IT side. This was truly a Merck group effort because we have actually colleagues performing similar pilots in life science and electronics. So we collaborated on this, and Ian and Mo were the representatives from TetraScience who actually did the lead engineering and project management on their side. And I'd like to take any questions.